It results in a lack of oxygen and nutrition to the body's organs, as well as a decreased exercise capacity due to an inefficient contractility state of the heart. And in patients with heart failure and animal models of heart failure, there's been suggested to be a role for contractile calcium. But before we can begin to understand what goes wrong in calcium handling, let's first look at what happens in a normal state. Calcium is a major player in the process of excitation contraction, or EC coupling. It's the process by which electrical impulses lead to mechanical movements in the cell. During EC couple, in the heart, RAR2 channels, or renin receptor type 2, it resides on the sarcoplasmic reticulum and is the primary channel for calcium release in the cell. During an active potential, voltage-gated calcium channels, or L-type calcium channels, lead to an influx of extracellular calcium ions into the cell, inducing calcium-induced calcium release on the renin receptor, leading to an efflux of calcium through the renin receptor from the SR. This calcium eventually leads to contraction on the actin-myosin bridges where calcium binds troponin. Upon relaxation in the diastolic phase of the heart, this calcium is removed from the actin-myosin bridges and taken back into the SR by the sarcoendoplasmic reticulum calcium ATPase, or SERCA2, and leads to calcium being residing in the SR for the next contraction. As we all know through studies of the last 40 years, cyclic AMP-dependent protein kinase A is the main hallmark stimulant of RAR2 at serine 2808, as stated in the fight or flight response in human beings. But in our lab recently has demonstrated that there's another site on RAR2 which can be phosphorylated, and this is serine 2814. This site is actually directly phosphorylated by calcium-dependent calmodulin kinase 2, or CAM kinase 2. This molecule has been shown to be upregulated in patients with heart failure and animal models of heart failure. But the problem is that it actually phosphorylates many downstream targets, including the L-type calcium channel and SERCA and other various targets. So the question I wanted to ask was, does this direct phosphorylation of CAM kinase on RAR2 lead to the development of heart failure? So to answer this question, we use two knock-in mouse models. These knock-in mouse models have one single amino acid change. The first model being a serine to aspartic acid, which is denoted the S2814B mouse. This mouse mimics the hyperphosphorylation of RAR2, leading to constant calcium leakage through the RAR2 channel. The other mutation is a serine to alanine mutation, or the S2814A mouse, which inhibits CAM kinase-mediated phosphorylation of RAR2. I first started with the S2814B mouse. Over time, in a long-term study through echocardiography, I evaluated that the D mouse show signs of cardiac dysfunction, indicated by an increased left ventricular posterior wall diameter during diastole, an increased end-diastolic diameter, and a decreased ejection fraction by 9 and 12 months of age. These results are also indicated by H&E staining on histological sections, in which you can see that the S2814B mouse's heart is very enlarged in comparison to the wild-type heart. These mice also show signs of increased spontaneous calcium transient after pacing individual cardiomyocytes and waiting for a pause. You can see that there's an increase in calcium leakage in these mice. So this was a subject of another study of mine, but for today we're going to focus more on the S2814A mouse, which is in a more clinically relevant state. So the hypothesis in these mice were that the inhibition of CAM kinase phosphorylation on RAR2 may prevent the development of severe cardiomyopathy in animal models of heart failure. To study CAM kinase phosphorylation on RAR2 in heart failure, use a particular model entitled transverse aortic constriction, or TAC. TAC is induced by subjecting a ligature, a 27-gauge constriction between the first and second truncus of the aorta between the right and left carotid arteries. This ligature induces pressure overload back onto the left ventricle, similar to a high blood pressure patient leading to cardiac failure. Over time, perform serial echocardiography, pressure volume loops, and tissue measurements, which we will get into in the next slides. So to actually check for CAM kinase phosphorylation of RAR2 in these mice, I use phospho-specific antibodies to check different levels of phosphorylation at different sites. In In the wild-type mice, you can see that there is a marked increase in phosphorylation at serine 2814, the CAM kinase site on RR2 after 8 weeks TAC, and more significant at 16 weeks post-TAC, whereas there's no phosphorylation in the S2014A mouse. As well, we also know that, as I stated previously, that the PKA site is very important on RR2. I also checked the phosphorylation of this site after TAC in both models, and it remains unchanged, although I am increasing the N in this group. Also, a hallmark of heart failure, as shown in very many previous studies by the labs, is the increase of the autophosphorylation or activation of CAM kinase itself. And I also see this increase in the wild-type TAC mouse, but I do not see any increase in the S2014A mouse after TAC. So to study this mutation in cardiac hypertrophy and failure, I wanted to look at the cardiac geometry and function of the heart. So echocardiographic analysis showed that after TAC, there was a similar hypertrophic response in both wild-type and S2014A mice. But at 12 weeks and 16 weeks, you see a start of a significant trend of the wild-type mice, leading to a reduction in their left ventricular posterior wall diameter during diastole. This is suggesting ventricular wall thinning due to cardiac dilation. And indeed, we do see an increase in end diastolic diameter in these wild-type TAC mice compared to the S14A TAC mice and the wild-type and S14A sham mice. To further study structural changes in these hearts, I once again used histological sections stained with hematoxylin and eosin staining, where you can actually see that both mouse hearts, have an increase in their left ventricular posterior wall diameter, but only the wild-type TAC has this overly increased size of their heart in terms of cardiac dimensions. This is also confirmed by post-mortem measurements using the heart weight to tibial length ratio, in which the wild-type sham compared to the wild-type TAC was very significant. And there was no change between the S2014A mouse hearts before and after TAC. So looking at cardiac performance, we can see that by using ejection fraction on echocardiography, we can see that the wild-type mice, we can see that both groups decline similarly up to 8 weeks, and we can see that after 8 weeks, we see this severe decrease in cardiac function in the wild-type TAC mice, whereas after 8 weeks, the S2014A mice have sustained function up to 16 weeks after the surgery. Noteworthy is that this time point actually correlates to the time point I showed you on the Western volume, to where the phosphorylation in the wild-type mice became significant at 8 weeks and beyond. Also, this was confirmed using chronic stimulation of the beta-adrenergic system using isoproterenol, which also showed similar results, but I don't have time to show you today. So upon inserting a catheter into the right carotid down into the left ventricle, we can also use this catheter to measure the maximum derivative pressure over time, which is a measure of contractility in the heart. We can see that the wild-type TAC mice display a depressed systolic function in their LV, whereas the S2014A mice have sustained cardiac function consistent with the ejection fraction data. Another hallmark in patients with heart failure is increased diastolic blood pressure. We actually see similar results in the wild-type TAC mice, where we have an increase in their end diastolic blood pressure. There is no change in the S2014A mice. So to further study, next we'll perform Mason's trichrome staining to evaluate whether there is any cardiac fibrosis in these mice, indicated by blue on the red tissue sections. So as you can see, both animal models actually increased in their fibrosis levels after TAC, but only the wild-type TAC was significant to its sham counterpart, and there was an almost significant trend between the S2014A mice and the wild-type TAC mice in terms of cardiac fibrosis. Also, looking at lung weight to tibial length ratios, you can see that the lung weight was increased in the wild-type TAC compared to the sham counterpart, as well as the S2014A mouse, suggesting that there was pulmonary edema and congestive heart failure. We can actually see that this is rescued in the S2014A mice. Another hallmark of heart failure in patients is markers of in the ventricle and also in the atria, either atrial natriuretic factor, or ANF, and brain natriuretic peptide, or BNP. ANF is in response to high blood pressure. The body uses it to compensate for these pressures.